So, Scvelo is the latest package to support RNA velocity analysis. In this video, I'm going to show you how I use Scvelo package in Jupyter Notebook and perform RNA velocity analysis. You can follow the installation guidance on Scvelo website to install Miniconda, Scvelo, other dependencies, and Jupyter Notebook. So now we can open Jupyter Notebook and do a demonstration with human fibroblast RNA velocity analysis. So following open the Jupyter Notebook, first step is to import Scvelo. Then we can just press run. So in next step, we need to load our data use SCV read function. So I prepared the data already for this demonstration. I did the cell clustering in Surat and prepared the NUM files using Cell Ranger and Venocyto packages. For beginners, the most difficult step is to prepare the correct dataset. Once you have the data, it is very easy to run velocity analysis, which I'm going to show you here. So we can use the SCV plot function to check the cell clusters before running velocity analysis. So I'm studying how resident fibroblast transit into myofibroblast in human fibrotic lung. So I want to use RNA velocity analysis and predict the origin of myofibroblast in fibrotic lung. Human lung fibroblasts are highly heterogeneous. For the purpose of this demonstration, I only use one cluster of resident fibroblast and one cluster of myofibroblast. Cells in cluster 0 on the left are myofibroblast, and the cells in cluster 1 on the right are resident non-fibroblast. So the image looks the same as my Surat analysis, so we can continue the velocity analysis. So for velocity analysis, we need to calculate the percentage of spliced and unspliced mRNA. Spliced and unspliced mRNA have been analyzed in previous step by Cell Ranger and Venocyto packages. Here, we just need to use the SCV plot function to plot the percentages. So as you can see, cluster 0 has 21% unspliced mRNA. Cluster 1 has 19% unspliced mRNA. So we then run basic data preprocessing uses field and normalized function for gene selection and cell normalization. We can press run. We also run the first and the second order moments. The second and the first order moments are needed for velocity analysis and the velocity estimation. So as you can see here, I run the data before. So it did not normalize the data. When you run your new data set, it will show the gene selection and cell normalization. So here comes the core analysis using Scvelo velocity tools. We can press run. So I'm running the stochastic model. You can also run the dynamical model. For dynamical model, you just need to change it here to dynamical, but you need to run recover dynamics tool first before you run the dynamical model. I'm not going to run the dynamical model here today because it takes a long time to run. So the velocity analysis is done. Then we can store the velocities using the velocity graph function in A data. So now we can project the velocity is in a low dimensional embedding using embedding embedding functions. We can press run again. So the embedding is done on single cell level. You can also embed the velocity as a grid and as stream. So my favorite projection is the stream one because it gives you a clear direction of the trajectories. Here you can see resident fibroblast transit into myofibroblast in human fibrotic lung. So sometimes the cell clusters are separate, they don't connect to each other. Then we can use the velocity graph function to visualize cell-cell connection between different clusters. So we can run again. So you can see the connections between cells and cells in two clusters. So we can also look at the velocity and the expression of individual genes. For example, collagen 1A1. So collagen 1A1 is a gene and that has a high expression in myofibroblast. As you can see here, we can run as many as function you like. For another example, we can identify genes that have a cluster-specific differential expression. We can run this function. Then here it will show 5 genes in each cluster. So as you can see from my demonstration, once you prepare a correct dataset, you can run all the analysis. For other code, you can just go to Scvelo website, check different functions of all the code. You can run the one you like. So I'm going to stop here. I hope this video is useful for your research.